Okay, well, it's my honor to introduce Dr. Danelle Haake from the National Great Rivers Research and Education Center in East Alton, Illinois. Dr. Danelle Haake began her career in ecology as a water quality manager for the Iowa Department of Natural Resources. She then worked as a restoration ecologist with the Missouri Botanical Garden. In 2020, Dr. Haake received her PhD from St. Louis University, where she studied how road salt impacts the diversity of invertebrates in urban streams. Since 2020, she has been the Illinois River Watch Director and Stream Ecologist with the National Great Rivers Research and Education Center. Today, she'll be talking about developments in community-driven science, people, salt, and mussel. We're delighted to have you. Please take it away.
All right, thank you. Over a little bit here. Okay. Yeah, thank you for that introduction. I'm glad to be here today to tell you a little bit about the work that I've been doing in cooperation with a large number of people who will be kind of recognized at various points during this presentation. Let's see. Make sure it's focused in the right spot. All right. So first I'm going to do a little bit of kind of housekeeping background information here, talking about citizen science and community science. These are terms that you may or may not be familiar with, but citizen science is kind of an umbrella term that has been used for 20 or 30 years to talk about any kind of involvement of the general public in scientific endeavors. So this could be, you know, it's some sort of voluntary participation, people doing science in their spare time. More recently, people have been kind of been using the term community science more frequently instead of the word citizen science, and I'll go into that in a second. But the original definition of community science has a lot more to do with the community part of citizen science. So community science being a scientific endeavor that is involving a community of people, so there's a specific location in mind, and the participants are learning about something locally and being able to take some sort of action. And so words are important. So understanding the implications of the language that we're using is important. I am no linguist, but we still have to, you know, have respect for words and what they mean to different people. There are a lot of people who don't like the term citizen science because it implies that you must be a citizen of a specific nation or locality in order to participate. To my knowledge, that has never been anyone's intention in using the word citizen in that context. But that's the way it can be interpreted. And so we want to, you know, citizen science is all about being inclusive. And so people are looking for a new term to use for that. A lot of people are trying to use the term community science for that. But again, words matter. And the term community science already had a meaning when people started trying to redirect the terms. So there's a lot of other types of citizen science type names out there. People talk about crowd science. Well, that's kind of a lot of people in one place at one time. That would be crowd science, but that doesn't apply to all of what I'm going to keep referring to as citizen science for today. Volunteer monitoring is another type of citizen science, another program under that umbrella, where usually you're talking about, you know, some citizen science might be just identifying whether or not something exists, but monitoring has some numeric quality to it. You know, you're collecting data points on the length of something or the size of something. So that's kind of a little bit of a background. Because I don't know of a more appropriate term, I'm going to continue using citizen science, meaning citizen of the world, not intending to exclude anyone. All right. So again, with citizen science as our umbrella term, I'm going to give an example of a few programs that fit under this umbrella in some way. The first is BioBlitz programs. BioBlitzes are programs where you have people coming together in one place at one time to identify generally anything that's alive. So it could be plant, animal, fungus, just a lot, identifying a variety of types of organisms in one place at one time. And then you can kind of compare that to a program like the Great Backyard Bird Count, which is a program where there's one time, but it's small groups or individuals that are doing monitoring throughout the world. There's other citizen science programs that are gamified. So you go somewhere on your computer by yourself and you play a game, and that game increases our scientific knowledge. This is actually done a lot with genetic research, and also there was another example, and it's kind of flown the coop for the moment. But there's different kinds of things that we're able to, because humans are better at pattern matching than computers. These citizen science programs that are gamified are very helpful for some research. And then two programs that I'll be talking about today are the Missouri Stream Team and Illinois River Watch, which are both water quality monitoring, volunteer monitoring program, where there's actually numeric measurements being made and reported. So those are all kind of the basic backdrop for what I'm actually talking about today, which is community-driven science. So you take community science and you go an extra step with that. What makes community-driven science different is that it involves a high level of initiative and a high level of engagement. So starting with the high level of initiative, the people who are creating this community-driven science project are just everyday people that have some sort of awareness of a local problem, a local need, something that they see is wrong and they want to get it fixed, but they need more information first. Now, being able to spot that there's a problem or a need means you have to have some level of prior knowledge or expertise. People don't just say, oh, wow, there's a dead zone in the Gulf of Mexico, I'm going to fix that today. Scientists don't do that. People on the street aren't going to do that. So you have to have an awareness of the problem and its scope and some idea of what needs to be done. So you take that awareness, and someone with initiative will seek an expert, someone who knows about this problem, and they find resources, whether that's financial resources or material resources, equipment. They seek those themselves. And then they co-develop questions and methods. And by co-developing, that can be just a group of individuals, a group of those citizen scientists, or those citizen scientists with those experts that they have sought out. So that's the initiative. That's what needs to be there to create a community-driven science program. But then once that program has been begun, there has to be a continued high level of engagement. First, by recruiting other volunteers. So these other volunteers may not be the leaders of the project, but usually a program like a community-driven science program is going to have other people brought on board to do the monitoring, to fill some of the roles needed in a project. They also will be collecting the data and managing that data. And then once the data is collected, they take part in whatever compilation, analysis of the data, and presenting what they found to their stakeholders, which, if it's themselves or their local city council, local media, or a broader audience. And even to the point of potentially writing reports and submitting scientific papers. So community-driven science is like other citizen science in that it involves the general public in getting science done, so to speak, but the citizens have a much greater role in the process. And so today, I'll be talking about two examples of citizen science, or community-driven science. But before I do that, and just by a show of hands, how many of you have participated in some sort of citizen science program? And by that, I mean, you've done science that wasn't part of your job, maybe not in your discipline, and possibly something that, most of us have gone out and helped a fellow researcher. That doesn't count. This is something you're doing it for fun. So, okay, several people here have done that. That's wonderful. How many of you have led some sort of citizen science project? maybe not been the leader, but have been involved in the planning process. Okay, very good. So yeah, I'm glad I'm not speaking to an audience that doesn't know at all what I'm talking about. And I'm glad that, there's a lot of scientists who, we still want to learn about things in other disciplines and, kind of tangentially related to what we do perhaps. Well, the first bit of work that I'm going to talk about is St. Louis Road Salt Project. So this is a project that started with a group of Missouri Stream Team volunteers who were volunteering together at the Litzinger Road Ecology Center. And just to kind of start off, I began as a Stream Team volunteer when I was an undergraduate and continued volunteering throughout my career. Not so much anymore. I do more work in Illinois now and don't have the time to do Illinois and Missouri. But we've, at the time that this began, I was a restoration ecologist with the Botanical Garden at the Litzinger Road Ecology Center. So part of my job was just kind of coordinating a group of volunteers, but really I was doing citizen science. We were going out, collecting data and contributing it to a citizen science database. So this group was going out monthly and doing water chemistry analysis. In January of 2011, we'd been doing monitoring for quite a while, but we noticed really high salt concentrations, really high chloride concentration at one of the sites we were monitoring. And by really high, normally it was, around 100. This measurement was around 3,000. So definitely something, and we measured several times to make sure it wasn't some kind of mistake. And no, it was actually around 3,000. Didn't take us long to think about it and say, oh, it snowed a couple of days ago. There's a bunch of road salt that was put upstream of here. I bet it's road salt because chloride is part of road salt. So the volunteers recognized together that this was a problem, that high chloride is a problem. Since it was something we were measuring as a water quality parameter, we were able to look in our resources and say, oh, this is actually toxic to aquatic life. So a group of volunteers, probably 5 or 6 of us, met with agency staff. And by that, I mean from Metropolitan St. Louis Sewer District and from Department of Conservation and Department of Natural Resources. And this was before Zoom was really a thing. So this was all in person. We convened this little meeting and talked about the problem. And basically, it's non-point source pollution. There wasn't much they could do about it. Well, as volunteers who are motivated by wanting happy water quality, we went ahead and said that's not good enough. So we started a regional volunteer monitoring project. Several of us knew other people who were doing the same monitoring in different rivers and streams in the St. Louis area. We got together, communicated with each other and said, hey, this is a problem, let's do something. So we began our little group. Ended up having 67 volunteers involved in the project over the course of about 10 years. And the first time we met, it was maybe 15 of us. We got out paper maps and drew on them and said where we were going to monitor, and then went out and did our thing. We came up with our methods, which basically we were using the Stream Team methods to monitor, except with some variations. We changed how frequently we would be doing it. Kind of set some parameters for ourselves. So annually, we would have meetings to kick off our season every November, which usually meant we met before the first snowfall. So we could catch, everybody would go out, do their sampling sometime in late November, and then go out monthly, weekly. People could go out as frequently or infrequently as they wanted. There was no set timeline. As the years went on, we had these meetings every year. And eventually, so we had, again, the Metropolitan Sewer District, Department of Natural Resources, Department of Conservation, as well as local public works directors would attend these meetings with us to talk about the problem, what we could do about it, what they were doing about it, and just making forward progress on the issue. So after a few years, we went ahead and put together some information on what we'd been finding. So this is just kind of a general overview of what we were finding in the streams. The size of the dot has to do with the watershed size, larger dot, larger watershed. And then the color scheme, the blues are in the okay range, the reds are in the not so okay range. Our highest concentration that was measured was about 8,000, which was the limit of the test strips that we were using. And then this figure was in this pamphlet here on the left. It was a little 4-pager that we put out, made available to, again, public works departments, to local community groups, anybody who was interested in road salt. So we put that together, shared our findings with the community, and the other was put together by MSD's Project Clear with our help. So that's the Stream Team logo on there because we were participating in getting this done. And then our volunteers distributed these to a lot of commercial parking lot managers and owners, you know, grocery stores, shopping centers, churches, hospitals, just spreading the information about how to do a better job. We also set up some educational booths because we wanted to educate not just the people who were doing the large areas, but also people who might be using it on their sidewalks. So we had these tables set up. There's invertebrates in that little pool in the center, and then explaining to people how much salt it takes to make that water toxic for the invertebrates. And then we had this little blue Christmas tree. Most booth opportunities, tabling opportunities like Earth Day and farmers markets, those are in the spring and summer. So in order to create a situation where they might actually remember this information when it became usable, we created these little ornaments and said, hey, take this home, put it with your holiday decorations, and then when you get them out in the winter, or in November or December, you'll be reminded of this information. And these were created by one of the other volunteers. They made these cute little drawings, and so the one side had the image on it, and the other side had the information on road salt and how people could help. And then the thing that I'm personally most proud of is beginning basically right when the pandemic started in early 2000, myself and 3 of the other volunteers, by that time I had my PhD, but these other 3 volunteers are retired professionals from other fields. One was a lawyer, another was a lab chemist, and another is a medical device developer. So people who aren't necessarily scientists, and certainly not aquatic scientists by trade. So the 4 of us met weekly and co-wrote a paper that has now been published. So basically what I'm going to talk about for the rest of this is what's in that paper. So this is the area that we were looking at. I mentioned earlier that we had 67 volunteers. They were working in it was 63 volunteers and 37. I'm getting my numbers backwards. 37 volunteers, 63 sites. That was the whole monitoring effort. What we included in this study was 31 of those sites because we wanted to make sure that we had consistent data as much as possible. So we had to have a certain number of samples collected in the winter and spring. And then also we were looking at sites that had aquatic invertebrate collections that had been done. So Missouri Stream Team has water chemistry monitoring. They also have aquatic invertebrate monitoring. And so we wanted to make sure that we included both data sets in this. So using GIS, we went ahead and identified some watershed variables for each of these 31 watersheds. We identified the amount of impervious surface and the percentage of the watershed that was road. Those coverages were provided by the sewer district. And then we also looked at the land cover maps to identify the amount of medium to high intensity road cover or high intensity development and the percent of undeveloped areas. So we analyzed these and a few other watershed variables to come up with and ran Pearson correlation to find out how much they were related to each other, because there's going to be a lot of kind of correlation between these because they're taken from the same data set. And urban areas, more urban is going to be like in one way, less urban will be the other. And the two least correlated variables were percent road and the percent of medium and high developed area. So we went ahead and we're using those as our two metrics of development. And that ended up being quite convenient because the percent road can be very well correlated, very well related to the amount of municipal salt application done on roads. So that we figured that's going to be a very closely related set of information. And then the percentage of medium and high intensity developed area will be closely related to the amount of the commercial contributions. Because if you look on the map, you can see that within the watershed areas that we're looking at, there's these lines that are those dark reds that are the medium and high intensity developed areas. Those correspond with larger roads like Lindbergh Boulevard and Manchester and Clayton. Well, those are commercial developments along there as opposed to the lighter colors that are more residential. Okay, so this is a summary of the data for our sites. All the way on the left, we're looking at sites that have the lowest percentage of roadway. And on the right, you have areas that have the highest percentage of roadway. You can see that there's, definitely a lot of information in here. And so I'm going to kind of break it down. So looking at, we looked at several different chloride metrics similar to the watershed metrics. And the two, again, that were least correlated with each other were the maximum concentration of chloride and the median concentration of chloride. So first, looking at maximum chloride, we ran a multiple regression of maximum chloride against the land use variables and found that there was a significant relationship between the maximum chloride and the percent road. There wasn't a relationship with the medium to high developed areas, which I'll probably just call development at this point. So that was a statistically significant relationship. And you can see that somewhere, the red dash on that line is the acute toxicity threshold set by EPA. That's 860 milligrams per liter. So, we don't really start exceeding that threshold in our data set until you get to somewhere in the 5 to 6% road surface in the watershed. Now, one thing I do want to mention as a caveat, these are grab samples that were collected. So it's difficult, none of these are going to be the actual maximum chloride concentration experienced in the stream. So in some cases, it might be much higher. It might be twice as high or even more than that because it's really hard to catch those peaks. Sometimes the peak concentration is only on the site for maybe 2 or 3 hours. Okay. So again, back to this. When we're looking at our median chloride concentration, the amount of road, the amount of development, and their interactions were all significant. And so what I found particularly fascinating was the interaction. And this is kind of maybe a graph, that type of graph many people have not seen before. So we're still looking at the median concentration of chloride on the y-axis. On the x-axis, we have road, and then we have the percentage of development, 1 standard deviation higher than the mean on the top and 1 standard deviation lower than the mean on the bottom. So basically, these are representing areas where you have more than average medium and high intensity development versus less than average development. So the one on the bottom, the dashed line, you can see, as you have lower percentage of road and you have lower than normal commercial development, you're not going to have much chloride. That's kind of what you would expect. And then as that percentage of road increases, the percentage of, or the amount of chloride increases. That's kind of the same pattern that we saw in the first graph, just looking at road and maximum chloride. But what's interesting is that when you have more commercial development, pretty much straight off the bat, you're going to have a high chloride concentration. And it's not quite a flat line, but within the confidence interval, you don't have a whole lot of increase in the chloride concentration as you add more road surface. And so the median concentration of chloride found in a stream seems to be much more associated with the amount of developed areas. So we're having more contributions to what I would consider the baseline, because take a step back, that as you have, the maximum concentration of chloride is going to be a representation of the short-term impacts of road salt application. Whereas the median concentration of chloride is going to be kind of the long-term impacts, because you have a system where you have a lot of peaks and declines. And so that median concentration is going to represent what's there on just about any given day. That's probably the mid to low concentrations that you find. And so going back to this figure, that means that as you have more commercial development, then you have a more long-term impact of that road salt on your community. Okay, so now we're going to move to the invertebrate response. So this is still the same figure we've been looking at. And one thing that you may have noticed is that those bars are different colors, and that it hasn't made any sense as to why they're different colors. Well, we'll change that in a second. All right, so this is the data for the aquatic invertebrate communities that were there, looking at a water quality rating, which is it's the method that Riverwatch uses to say how much diversity is in the stream. So things that are sensitive to pollution receive 3 points. Things that are moderately sensitive to pollution, you get 2 points. And things that are tolerant of pollution, you get 1 point. So aquatic worms, leeches, those are 1 point. Mayflies, caddisflies, stoneflies, those are each 3 points. So the more points you accumulate in your sampling, the better quality your stream is going to be. So the colored codes here are the stream team's red for poor water quality. The tan is the fair quality. The light blue is good water quality. There's also an excellent category. None of our sites reached the excellent threshold. But looking at this, so again, we have our percent road increasing as we go up the site. So now that you know what those colors mean, kind of looking at the figure on the top, and if there at any sites where you have about 25%, maybe more like 20% of the sample is at or above the acute toxicity threshold, that red dashed line, all of those sites are in the poor category. By the same token, all of the sites that are in light blue, the sites that are considered good quality, have a median that is at or below the chronic toxicity level. So you can see that we're definitely just kind of eyeballing it from this. There is some effect of the road salt on the invertebrates, or it seems that there should be. But then what about these 4 sites here at the low end? These are places that don't have a whole lot of road surface. They don't really have a strong chloride signature, and yet we have some fairly poor water quality ratings. Well, unfortunately, we live in a complex world, so we're not looking at necessarily the effects of road salt here. There's a lot of other stressors that could be causing this sort of response. And so what we did is we used something called Quantile regression to help tease out this response. So in this, the figure that's shown here, you have an organism is experiencing an environment. Let's say the measured factor is the amount of food in a system, and the organism response is the growth. So as there's more food, you have more growth. You have a positive response. In a perfect world, you get a nice linear relationship like this. You do your mean regression and you get a number and everybody's happy. The real world, like I said, is a little bit more messy than that. So if you take that same system and you add some unmeasured contributing factor or unknown contributing factor, the more contributing factors you have, the more and more of your data is going to be below what the normal or what the mean regression would be. And so what we can do to kind of compensate for that is to measure where that line would be, which would be measuring a certain quantile. So measuring a percentile that's not the mean. We're not looking at the middle of the data. We want to look at the upper edge of the data. So the 90th, the 95th, or 99th percentile of the data is what really is telling us what that threshold is. Because otherwise, if we do a mean regression, we're going to end up with a line like that, which doesn't actually tell us how the organism is responding to the factor that we're measuring. So for our work, we looked at the quantile regression at both the 90th and the 95th percentile. We looked at 2 of them just to kind of make sure that we weren't getting some artifact of some data being weighted heavier than others. And so looking at the invertebrate responses, what we find is that we do have a system that looks like it probably, that the quantile regression is giving us some information here. So if we start at the top, looking at the impact of maximum and median chloride on the number of animals that were collected in a sampling event. We do have, kind of a response there where it looks like, once you get a maximum chloride concentration of around 3,000, you're losing about half of your animals from a sample or from a site. Median chloride is not quite as strong of a response, but still, again, you're losing half of your diversity when you get to about 600 milligrams per liter as your median chloride level. The relationship becomes even stronger once we take into account the amount of time the volunteers spent collecting those invertebrates. So that's the catch per unit effort, the number of animals that were collected per minute. And here again, you can see that around 3,000, you lose about half of the animals collected. And for the median chloride, you have an even stronger relationship at something like 300 milligrams per liter of chloride. You've again lost about half of the animals. So, and then the next set down, looking at the water quality rating, again, we're losing types of invertebrates as well. Gradual decline in the types that we're finding fewer of the sensitive organisms as we get into higher chloride ratings. So we're decreasing in both abundance and diversity. Okay, so kind of back to the point of some of this. This is a community-driven science project. That scientific paper that I just described to you, that was written completely with the input of those volunteers. Yes, I was the one who ran the statistics, but they learned about the statistics and they agreed with what they meant. Sometimes we would see something and be like, okay, what does this mean? And they would explain, they would be able to understand what the data meant. So, they read research papers, they wrote sections of the paper. Yeah, everyone contributed to pretty much every part of it. One of the volunteers did some of the GIS work for it. So full contributors in the paper. It's not just me slapping their names on it. They worked hard on this and they did a great job. All right, so high level of initiative, high level of engagement. Again, initiative, they got check boxes on all of these components. I started out as one of the initiators. I didn't initiate it by myself. It was definitely a team effort. But at the time, I was doing this as a community scientist or citizen scientist. This wasn't part of my job. This was my free time. High level of engagement, collect and manage data, presenting findings to stakeholders in the community, and writing reports and scientific papers. So this is what community-driven science is all about. So that's the first example. I'm not done yet, but I want to acknowledge the people who participated in that project first. So this is a list of the volunteers. The 3 that are in bold are the ones who helped to write the paper. The Missouri Stream Team staff, which includes people from the Department of Conservation and the Department of Natural Resources. MSD, Public Works departments, speakers came out and spoke to our group at our annual chloride kickoff meetings from both SLU and WashU. And then that's some information on the publication. I'd be glad to share more information on that later if you'd like as well. All right. So that's where we're at from salt. I'm going to move on to freshwater mussels. So for the first story, I was the citizen scientist. In the second story, this is happening in my role as the director of Illinois River Watch. So I'm not the citizen scientist who kicked this off. I'm the person they came to get the resources. So the real hero of the story is the Upper Sangamon River Conservancy. So this is an all-volunteer group that does monitoring and recreation and other work and cleanup work on the Upper Sangamon River, which is located they're centered in Mahomet, Illinois, which is very close to Champaign-Urbana. So they did their first mussel surveys in 2011. They have 4 sites that they started out with. 3 of them they would monitor on a rotational basis. So they'd monitor site 1 in year 1, site 2 in year 2, 3 in year 3, and then in year 4 they'd go back to site 1 again. So there was some repeat monitoring, but they didn't want to overexert an influence on the mussel community by digging them up too often. But there was a 4th site that they added that they were doing not every year, but they would do for a few consecutive years and then give it a rest for a year or 2 and then do a couple years in a row again. So there's 4 locations that they've been working at. And their initial questions, they were interested in understanding, there was a wastewater treatment plant in Mahomet, and they were concerned about the impacts that the effluent might be having on these sensitive freshwater mussels. In 2014, they did do an upgrade. They completed an upgrade to that wastewater facility. And so that became a little bit less of a concern, but they were still interested in continuing the project. But they wanted to do more. So they came to me and asking for support in 2021 in order to turn this into a tagging project. Because they're collecting mussels, but they never know if they collect the same one twice. And so they wanted to tag, find out, and answer a few other questions about what they've been finding. So what they had been doing, like the people in this picture, they would get into the stream, they'd have a mesh bag, and they just dig around in the sediment with their hands to find these mussels. They'd be sitting in the water, crawling in the water, sometimes standing and hunching over, but you can only do that for so long. And, usually you'd be in an area for a half hour or more at a time. So, everybody would get in the water, get mucky and get wet. It's kind of fun. So they would collect these mussels. They take them to a station that they set up on the bank, take them to a table. They would measure the length. They would measure the depth of the shells, and then they would return them to the water, or after they'd identify into species. So they have this, 10 years of information, and they didn't really know what they could do with it, especially because they didn't have any way of knowing if any were recaptured. So they couldn't really estimate a population size. So that's one of the things that they really wanted to do. So in a kind of a collaborative process, working with myself and then also working with Sarah Douglas of the Illinois Natural History Survey, who is a malacologist out there, we came up with a plan, again, with a lot of their input. So they selected 8 sites that they wanted to monitoring them in alternating years. So in 2021, we did the first 4 sites. 2022, this year, we did the other 4 sites. So we'll have our first recapture year next year. We created and they designated repeatable monitoring zone with transects. So we have 10-meter transects at each location. And there's some permanent markers that have been put in place so that in future years, we can go back and revisit those same transects and know which mussels were taken from which locations in which years. They standardized the amount of collection time committed to each transect. Each transect is monitored is sampled for 90 minutes. All of the individuals are then brought to shore, kept in numbered bins, so we know where they came from, and then they're tagged, and then put back in the same transect that they were taken from. So we can, they'll be able to potentially, if we find that there's certain species that are moving up and downstream, that will be interesting information. We also began measuring some new parameters. We took some sediment collections from the sites to see if that's maybe a parameter that's influencing something about the species. The temperature, and we have temperature conductivity loggers that are in the stream, and we're taking the mass of the mussels as well. So these are some of the additions and changes we made, just kind of standardizing things. Again, the citizen scientists had complete input on all all stages of this process. And one thing that was really important to them was that they wanted the program to continue accommodating kids. They have a lot of families that will come out to do the monitoring. They have a lot of scout troops that come out and do monitoring, Boy Scouts, Girl Scouts, 4-H, all sorts of groups that involve children. And that's really important to them. So in order to involve those groups and still have a more scientifically reliable data set, if we're standardizing the amount of collection time that's committed to these areas, what we did is created a separate downstream monitoring zone. So in addition to the 100 meters we have where most of the work is being done and where all of those mussels are tagged, we have a downstream, we have 2 50-meter zones that are a little bit more of a free-for-all. You take your kids in there and you do the monitoring, and if you monitor for 30 minutes, that's fine. If you're in there for an hour, that's fine. Just tell us how long you're there. That's sort of a setup. And that's actually going to be really beneficial in the project, because if we have tagged mussels in the upstream area and over the course of the 2, 4, 6, 8 years, those migrate downstream due to storm or just due to their own movement, we wouldn't find them, except now there's a possibility we'll find them because we have this area downstream being sampled as well. All right, so another addition that we've made to the project is we have these little photo booths. And so for every mussel that's collected in the 100-meter primary area, all of those are tagged. And they have their picture taken from above and from the side. And these are intended to be repeatable photographs so that we can have a better indication of, if we recapture a mussel, how much is it growing and potentially where is it growing. So there's plans to do some digital morphometrics using these photos. Another example, these are all pictures that were taken at different sites on different days. So having repeatability in the appearance of the photos has been, I think, will be very helpful. But you can see we're getting lots of different shapes and sizes here. So there's going to be some interesting work done, hopefully, on this. Another thing that we added, and both the morphometrics and this genetic work that we're doing, some tissue samples were collected from 2 species. This and a lot of the work done with this project is actually funded with the Living Earth Collaborative. So definitely want to give a shout out for that. But so this is Sarah Douglas here collecting a tissue sample from one of the mussels. And those are in the process right now of actually being sequenced. But again, this is citizen science. And so citizen science includes an education component. And so we've had a lot of volunteers come through this program. It's led by citizen scientists, but it's also, and those people are very involved in the process of the community-driven science part. But then there's also just people who come and participate. And we've had anywhere between 20 to I think 100 people participating in any given event. So we can accommodate some pretty big groups at some of these sites, but education is always an important component of making sure, partly just so people understand why mussels are important, and also so people can, we help them learn a little bit about identification, about the importance of different types of habitats, a little bit of everything. And while this is kind of an ongoing project, and we just finished our collections for the year a couple of months ago, so we haven't done really any official analysis yet, but I can give you a little bit of kind of our what we've been looking at the data and looking, what we're seeing and what we might be looking at in the future. So this is a map of the 8 sites, and you can see that little star there that's turning. That's where that wastewater treatment plant is. So it's kind of, we've got 4 sites upstream and 4 sites downstream of that. In the data, the Sangamon River Forest Preserve, the one on the left, is our furthest upstream location. And there's 3 species, fat mucket, monkey face, and spike. Those 3 species were only found at that site. Like I said, that's our furthest upstream site. You'll also notice that we had over 400 mussels that we tagged in that location. At all of the other locations, we had between 100 and 220. So that site had more than double what we found at most of the other locations. Now, and since they're tagged, that means that was in the stretch, the 100-meter stretch. That's not including anything that's downstream. So this is all with standardized sampling times that we're finding that many more in that location. And one of those species that we found, the spike, that is a state-listed threatened species. And that's the first time in, I think, 30 or more years that species has been found in this watershed. And that individual actually was found in the downstream part by a Boy Scout. So, you know, children contributing important information, not just like a little bit of something. That's the first time that's been found in 30 or 40 years. So that was really cool. And then the other thing that we noticed is that there's 2 species that were only found at the 3 downstream sites, and they were both found at all 3 of those sites. So there's, you know, we feel like there's something going on there, something that we're going to look into more in depth. And overall, we did find almost 2,800 mussels of 19 species, and a little over half of those are the ones that we've tagged. So we've got over 1,400 mussels out there waiting to be recaptured, hopefully. All right, so one of the things that we were wondering if it might be kind of triggering that difference in what we're finding upstream versus downstream is the type of sediment. You know, the habitat is going to have an influence on what's living there. But as we kind of looked at it more closely, I mean, the Sangamon River Forest Preserve, we had some samples collected there had a lot of gravel, and some only had a little gravel. So maybe it's the fact that there's a highly variable sediment substrate available to them. So maybe that's why there's more species there and more mussels in general. But then looking at the 3 downstream sites, they're kind of all over the place. You know, the Shady Rest is a much more gravelly site. Lodge Park has almost no gravel and it's mostly sand. And then Allerton is mostly even finer sand. So again, this is just kind of like our first stab at looking at the data to see what might be there. And it's looking, you know, without doing anything statistical, it doesn't look like sediment has a lot to do with which species we're finding where at this point. There's a lot more we could do with that though. With the genetic work, these are the 2 species of mussels that we're looking at. And that, like I mentioned, we have samples currently being sequenced. The tissue samples, like I mentioned, were collected by Sarah Douglas of the Natural History Survey. And then I have an intern, Josh Turner, who has been processing the samples. We got them sent off now. Next semester, he'll be doing kind of the follow-up on that. And he's been working in Christy Edwards' lab at the Botanical Gardens. So they're a very helpful partner in this. And I think they're kind of enjoying, you know, the change of, like, normally they're obviously working with plants. So it's a little out of their normal wheelhouse to be looking at animal genetics. So I hope they're enjoying that. All right, and as far as the outreach component of what's going on in this community-driven science project, first on the left there, we have an ArcGIS story map that's in the process of being generated. There's, you know, it's a fairly complete draft, but there's still some information that needs to be put together before it's released. So that will probably be coming out in January. And then also in January, on Tuesday, January 17th, we're going to have the film premiere of a short 6.5-minute film that was put together during the 4 sampling events this year. We had a film crew out who was filming the whole process and interviewing people, and they had a drone out getting footage, and it's just and I've been able to kind of follow the process of the editing for the film, and I'm really excited about the release. So I hope if you have any interest at all in mussels or community science or citizen science, I hope you'll join us for that. There'll be a live event at National Great Rivers Research and Education Center starting at 6, and then the film will be played at 7. Or if you don't want to trek up to Alton, you're welcome to join. It's also on Zoom. So, all right. So again, how are we doing on this community-driven science thing? You know, we definitely have the initiative. The people at the Upper Sangamon River Conservancy have done a great job seeking experts, seeking resources. We've had 2 grants to fund the project. The first was the Living Earth Collaborative grant to kind of kick things off. And then the film is being funded by a grant from Quasi, not entirely sure how they pronounce their acronym, but their name is even longer. So as far as the level of engagement, you know, they've been collecting the data. You know, almost all of the data has been, I mean, there's only 3 people with master's degrees, associate masters or PhDs related to water science who are showing up at these events. And there's a lot of data being collected. So it's almost exclusively being collected by the community scientists. Writing reports and papers, haven't gotten there yet, but this is only year 2. We haven't had a recapture season yet. So we're looking forward to, I'm sure that's just a matter of time before that comes as well. Okay, so on this project, again, want to acknowledge the partners. One that I haven't mentioned yet is the St. Louis Zoo. They're going to be doing, they're working on adding some mussels to their insectarium, I think. So I'm not quite sure how insect mussels, but that's okay. It's a zoo and do what they want. But they want to have a freshwater mussel program there at the zoo. And so they're kind of contributing through the Living Earth Collaborative, and we're working with them on that. So almost done. I just want to kind of bring up a couple of common threads that I hope you noticed through these 2 stories. So there's contributions that have been made by the programs that are kind of the initiators of the citizen science. They provide this baseline training, informing the volunteers, educating them, and giving them the tools to not just collect the data, but as they think about it, they can ask their own questions. The programs develop individual relationships with, you know, I know the names of the people at the Missouri Stream Team, the people at Upper Sangamon River Conservancy know who I am. You know, it's not just people talking to an organization, it's people talking to people. The programs made additional resources available. They asked for tags. I was like, let's find you some tags. The stream team made an abundance of chloride test strips available to us to get the work done as well. And then the programs recognize the efforts that the citizen scientists are doing, saying, wow, these people are doing great. Let's put it in our newsletter. Let's talk about it to these other stakeholders, making sure that the word gets out. And then the volunteers, they participated in these existing programs for years before they kind of took on these leadership roles. This isn't the sort of thing that you try it once and then you're like, okay, I'm suddenly going to take charge and become the leader of a massive undertaking. And then so they participated and then they took on leadership in this local issue that they had. They identified a question or a concern, something they just wanted to know more about. They connected with the different collaborators that were available and then engaged the local community in the efforts as well, either as volunteers or as people who should just be interested in what's going on because it's in their community. And then with those together, then I'd say we have had 2 very successful programs. And with that, I would be glad to take any questions that you might have.
Okay, so you know we don't have a microphone available, so speak up if you have questions so we can hear it.
Danelle, if you can just repeat the question. That would be good.
I can do that.
I have 2 questions. The first one is, how do you get the word out about these community science programs? Like, how are you reaching these non-scientist communities?
Okay, so the question is about how to reach out to the citizen science or to the general population to encourage them to become citizen scientists. A lot of it's word of mouth, people who we already know asking their neighbors, or people who are participating in other organizations, for example, the Master Naturalist Program or scouting groups or through their schools. Those are all good ways, but we've also had a lot of success in using press releases. So one thing that I hadn't mentioned is that as the director of Riverwatch, I've started a chloride monitoring program for the state of Illinois. And they've, so last year was our first year. We're in the beginning part of our second season of that program. I had 2 training sessions and we had a press release on those, but there was a newspaper up in the Chicago area that's like, hey, we want to do a story on this. But then by the time they were ready to run the story, both training sessions had passed. So it's like, hey, I'll do another training session, just get it in the newspaper. And they publicized it. And within 2 hours, I think I had 40 people registered for it. I think now it's up to 50 people. So if anybody, you don't even have to be in Illinois. If you're interested in chloride monitoring, feel free to sign up for that as well. It's on December 7th at 7 p.m. So always got to plug it. See, plug it anywhere you go.
My second question was about tagging mussels. I'm curious how you do that, and if those tags are more like you've caught this one before, or if you can recognize individuals after 2 weeks.
Okay, so the question is about the process of tagging a mussel, kind of basically what's it look like, and will we be able to identify the same mussel again? So here's a picture of a mussel tag being put on. You can kind of see it's what's called a hull print tag. Most times you do some kind of tagging, they give you one tag that has a number on it because we're working with bivalves, and if one of them happens to die, it has 2 shells, so we want a number on each shell. So if we only find 1/2 of the shell, we'll know which one it was. They have a 4-character code on them. Ours are just numbered 4 zeros up to 5,000. We have 5,000 tags, or 4,999, I guess. So there's 2 with the same number. So each animal, you can kind of see it has a tag on each valve. And then we should be able to go back. And then if you look at these, so the number on that's written on that tag, the 2013, that's the number that's on the little hull print tag that's on the organism. That way, because otherwise we wouldn't know which animal goes with which picture, because you can't usually read them in the pictures very well, just based on angles and things like that.
Thank you.
Sure.
Any other questions?
So how possible that you can put a very small chip on each one so that you don't need to take them out to monitor the certain, for example, like a drone, just survey their water surface and to get the signals from
Right.
Yeah. That's a lot of time.
Yeah. So the question is, would it be possible, instead of putting a tag on them, to put like some kind of microchip, in order to, instead of having to disturb them every time you want to find them, you can just use some kind of sensor above the water to then identify them. I think that's theoretically possible. I've kind of looked into it a little bit, but we've tagged already, like I said, over 1,400 mussels. So you certainly couldn't do, at least I don't think we could afford, unless we found a massive grant, I don't think we could afford that. But I have been kind of looking into, if we find, with our first set of recaptures, if we find that there's certain species that move a lot, that maybe we can go back and do a separate, smaller study where we would have members of that species tagged. And then, or with something like that, like radio telemetry sort of thing, and see how much they move from week to week. So yeah, it is possible. It's similar to what you would use for fish. Some of them probably don't work as well through water, so we'd have to be careful about that. But yeah, we would, just like the tags, we would just basically use super glue to glue it on the outside of the shell. Yeah.
Well, I have a couple questions.
Sure.
A really simple one. I was trying to think, you have these different salt concentrations. I remember the numbers, 100, 800 or something, I think was milligrams per liter.
Right.
Just as a reference, what is the salt concentration of like a marine system?
Okay,
I
yeah.
was just trying to figure out what that fit.
Right. Okay. So the question is about like salt concentrations and what is a range of, like, what would you find freshwater? What would you find in the ocean? So relatively pristine streams should have a concentration somewhere on the order of 10, 20, maybe 30 milligrams per liter. And that would all depend on geology because we naturally find salts in our soils. Our EPA guidance is 230 milligrams per liter is the chronic toxicity level. 860 is the acute toxicity level. And what you find in the ocean is about 20,000. Now I had some data loggers out as part of my research, and there was one site where I had concentrations that were more than you would find in the ocean for a period of about 3 days. And so that was something I was fortunately, thanks to Google Earth, was able to link back to an uncovered salt pile on a parking lot a quarter mile away. So that's kind of why that work that some of those citizen scientists were doing is, reaching out saying, hey, cover your salt piles, spread it thinly. You don't have to have a thick layer of salt down. You just put a few grains out at a time, So.
And then my second question, imagine, there's two main functions about citizen science. One is to collect more data, data that's hard for you to do or hard to pay a lot of people to do. The other is to educate the public by getting them involved. For this project, how would you weigh the relative proportions of those, and what goes into your thinking when you're making a project?
Okay, so yeah, the question is the two primary focuses of citizen science being, the data or research versus the education component, and kind of what is the comparison in these projects and maybe some other ones, and how do you go about picking projects that way? So in these projects, the process that, even for the leaders of the chloride project, there was a lot of education going on, not least of which for myself, because that project is what kind of got me interested. We were doing that for 5 years, and then I decided, what? I want to learn more. I want to do more. I'm going to do a PhD on road salt, So there's definitely a component. There's always an education component to citizen science, even for an expert. You're always learning something, because that's actually part of the purpose of research is education and learning. So the education is always going to be a strong component and should be. But the education is kind of dependent on the citizen scientists themselves. Do they want to learn something new, or do they just want to use what they already have? Or in the case of some of like the gamified citizen science, do they just want to move their mouse around and play a game? So a lot of this is about meeting people where they are and making sure that you have something. Some people want to do something complicated, that they have to be able to identify things that look similar. Other people don't want to go that far. And so, like, for example, in Illinois River Watch, we have the mussel monitoring, we have regular invertebrate monitoring, and we also have the chloride program. For chloride, it's a 1-hour training session. And then you go out and you stick a test strip in the water and you have a result in 15 minutes. For the traditional invertebrate monitoring, it's an all-day training session. And you have, then you go out and spend like 3 hours collecting a sample. And then you spend another hour or two looking at it under a microscope. And then you send that in. And so there's a lot of levels of involvement. And so I think it's most important is giving people options to be as interested as they want.
I guess in your case, were you motivated by scientific questions and then you wanted to get help using people, or did you feel like your mission was to get people involved in water quality issues and then you thought of a project that would do it? So for myself, and I'll use the salt project as an example, I was interested in water quality. That was where my bachelor's degree had focused. In my role at that point, I was working more with plants and ecosystems as a whole rather than streams. And so doing the stream work was kind of like, I get to play in the water and I love it. But then I'm also kind of an environmentalist and something of an advocate at heart. I may or may not do that as part of my work, but in my personal life, it's like, hey, this is a problem. It shouldn't be like this.
how do we fix it? And so that was my inspiration for the road salt project, is that, you know, we can't take these freshwater ecosystems and let them become oceanic or estuary like. You know, we have to allow these animals to live in conditions that are actually something that they can handle. So trying to find, you know, first finding the scope of the problem, and then finding out, okay, what can we do to improve it? And so that the work that I did for my PhD, one of the projects was to look at how the use of salt brine instead of standard rock salt, how that influences how much salt is reaching a stream. You know, brining is considered a best management practice. Does it actually work in residential areas? No one had looked at that yet. And so that was what inspired me to do that as part of my PhD, was to find out, you know, what can we do to make it better? And that was something that is effective. Yeah.
Okay, well, I think we're out of time. If you have more questions, we'll be happy to serve you food just across the hall. And we'll give one last round of applause for our speaker.
Thank you.